Next up, we have gut-brain axis therapeutics, and had a chance to briefly meet Rosie before, so I know it's Rosie and not Rosa. So Rosie.
Good afternoon. I am Rosa Krajmainik-Brown, or Rosie, and I am a co-founder of gut-brain axis therapeutics. We are a company that does microbiota transplant to help heal the gut and the brain for autism and Pitt-Hopkins. Pitt-Hopkins is a rare genetic disease. You probably haven't heard about it. Our vision is to heal the gut and heal the brain. Autism diagnosis is increasing rapidly in our country. The latest numbers from the CDC are 1 in 36 children, school children. 40% of children and adults have gastrointestinal symptoms, and we know that worse gut problems lead to worse autism symptoms too, or they're correlated at least. There's no FDA-approved medication for core symptoms of autism in the market yet. What is our product? Our product is a therapy that includes highly purified bacteria, gut bacteria, from carefully screened, very healthy donors. You can think about it as a super probiotic. A normal probiotic normally has 1 to 10 species, and this probiotic has more than 500 species that actually are going to their nature environment, which is the gut. We were the first team that showed on humans, on clinical trials, that we can change the microbiome for kids with autism using this microbiota transfer therapy and reduce gastrointestinal symptoms and also reduce some of autism symptoms. So here I'm showing you some of the data that we published on our phase 1 trial. On the left, you can see a graph where we show GSRS, which is a measurement of GI symptoms. The higher the number, the worse the GI symptoms. If you see a number 1, that's like a healthy gut. You can see that in the beginning of our trial, most of our children had a lot of GI symptoms. Our treatment was 10 weeks. At the end of the treatment, many of those GI symptoms were reduced dramatically. We followed up on these children 8 weeks later and 2 years later. You can see that at least on the GI symptoms, some of the benefits were lost, but there's huge improvement and huge gains even after 2 years. On the right side, you can see that our ASD symptoms, also autism symptoms, improved dramatically. In the beginning of our trial, you can see first the red bar. The red bar is the percentage of the children in our trial that were in the severe category. You can see that red bar coming down drastically at our follow-ups 8 weeks and 2 years later. And many of our children improved to a point where they were considered either just on the mild to moderate symptoms, and more than half of the children were in that category, and almost half of them were in a category where they had minimal to no symptoms reported. We also had every participant stayed on the trial, no dropouts, which is amazing on a clinical trial, and a follow-up 2 years later where we had all 18 participants. This tells you about the interest of the community and the positive success of the trial. FDA granted fast track status for an entity for autism based on these results. We just finished running a phase 2 trial with adults and also a phase 2 trial with Pitt-Hopkins, and we have some exciting results on that. We have a large market for Pitt-Hopkins. There's no FDA-approved treatment for Pitt-Hopkins, no FDA-approved treatment for symptoms of autism, only 2 medications that sedate children, likely to be the first FDA-approved medication for autism and core symptoms of Pitt-Hopkins. Potential sales, about $50 million per year for Pitt-Hopkins, but we have the potential of getting $100 million voucher for a rare disease. And autism, $1 to $2 billion per year. This also has the potential to treat many other gastrointestinal and gut-brain disorders like ulcerative colitis, Crohn's disease, Parkinson's, anxiety, depression, chronic fatigue, and maybe Alzheimer's. We have a great team, and we're ready to execute. Dr. Jim Adams and I were the first one, as I mentioned, to do this clinical trial with kids with autism, and we're experts in autism and the microbiome. He is the father of an adult with autism, and we have Alex and Mike, who are on the production team at the University of Minnesota. And with this, I conclude my presentation, and I would be happy to take any questions.
I have a question on the 500 plus
The
species that you've selected as part of your treatment. So how big was the sample of the super healthy individuals that you identified?
So we don't select species. The microbes come from healthy donors, but we purify the microbes, but we don't select species. That's not something we do yet, because we do not know which species to select for. Oh, so it's a very diverse community, and the idea is that when you put a very diverse community, you will increase diversity, which is one of the things that we wanted. It's a very diverse community of microbes that are, most of them, beneficial microbes. And we actually recorded that the microbiome changed. We increased diversity, and we increased the relative abundance of many beneficial microbes, for example, bifidobacteria, in our trials.
So how do you envision manufacturing your product long-term and the scalability, I guess, of your product?
That is a great question. So Mike and Alex, who are our collaborators in Minnesota, are in charge of the production. They have a donor program where they screen really carefully all their donors, and they actually worked with, I don't know if you guys know OpenBiome, which was a company that was actually providing FMT to patients in hospitals for Clostridium difficile. So it'll take some effort to scale up, and that's part of our plan, but we have a plan. In the future, maybe 10 or 20 years, it would be great if we can identify microbes, which I don't think it's one, it's probably going to be a team that we can grow in a fermenter in a bioreactor. In my center, in my lab. Yeah.
And how is you're familiar with Ceres, the company that just approved for?
Yes.
So how does your manufacturing is different from them? Can they provide a roadmap for what you are trying to do? Because they are also using stool to...
Yeah, so I get confused. There's two companies, Ceres and another one, and one of them actually uses spores, and I don't remember if that's Ceres. It could be that that's Ceres, and if they are, so that's the other one.
No, Ceres is the one that actually
so their manufacturing could be a guide for us too. I think, if I remember correctly, they do an enema, so that's one of the differences of their product and our product. Our product is not an enema.
Okay. No, it's not an enema. It's actually, it was just approved. It's an oral product that... same thing, uses human...
Is it a pill? Is it a pill?
It's a pill. It's an oral. It was just approved a few weeks ago. Not for autism.
Not for autism, for Clostridium difficile. Yeah, for C. diff. Yeah, and so the other part of our IP is that it's not just the pill, it's a whole treatment. So the treatment therapy for the way we have it and the way we have the IP it's a protocol. It's not just the pill, it's a protocol that you need to follow. And it's very different because for C. diff, you take one inoculation and that's it. For autism, we had to do a treatment for 10 weeks where we had a high dose initially and then a low dose daily. And we had an antibiotic before. And so it's a protocol. It's different. The product is different.
Have you talked with Nestle?
To who?
Nestle. They invested 150 million in that other company. Yeah.
That's good advice.
Yeah, you should.
Thank you.
This is very interesting. Congratulations.
Thank you very much. That's a great presentation. So we're an investor in the other company that you mentioned, Finch Therapeutics. So being in them, we understand the challenge as it comes to run a clinical staged microbiome company, especially in this market. Could you talk me through your financing status and how much you raised so far, et cetera?
Yeah.
Because as we know in this space, it's like usually science don't fail, you just run out of money.
Yeah. So we did our phase 1 with about 700K. We had 18 participants. Phase 2 for our adults, we had about $1.5 million for 56 participants. Phase 2 for our children, we had about 1.3 million for 50 participants. And for Pitt Hopkins, that was a pretty cheap study. So Jim and I have done more than 10 years of research on this. We have several patents. We've done 4 clinical trials. This is about 3.7 million that have been invested on our side. And the UMIN team also has invested about 4.5 million. So there's been about 8 million invested in our technology already. And what we're looking for right now is for Pitt Hopkins, which we already have the designation of rare disease, an orphan disease. We already have that. We're looking for drug approval, which would be great if we get it. For Pitt Hopkins, where we need an investment of about $1 million, we already have $750K offered or vouched. For autism, we need an investment right now between 1.5 and 3 million for now to move forward. So our next step would be dosing studies, phase 2 dosing studies. By the way, we're in touch with Mark at Finch. We're still in touch with Finch, working with them. So our next step is for autism is dosing studies, and hopefully that will give us enough information to move to phase 3 trials and drug approval. That's what we would like.
Thank you so much for that.
How is this affecting the behavior change in people
I know you
with
guys would
autism?
ask, so I have this. Okay, so here are some of the things that we saw that were changing in the behavior of the children after the treatment. And so this is what we saw in our phase 1, and some of these things is what we're looking at also in our phase 2. So the children are less irritable, they have better communication, their cognition improves also, their hyperactivity improves, and so there's many different things that we measure, and so that's the behavior part that is improving.
And how old were the children that were in this
For
trial?
this study, they were between 7 and 17.
7 and 17, and
Mm-hmm.
how does,
We couldn't treat them, we couldn't treat younger children because we had an IND and FDA said, hey, not very young children are not allowed in your trial.
but the question is, does this, like, because you don't have a placebo here,
We don't have a placebo. This
how
was
does
open.
this, like, how is this different than the natural variability in these endpoints as well as the natural progression?
That's a great question. So we didn't have a placebo there. My collaborator has run some other trials, and he said, I've never saw the effect to be as big as we have. We just finished our phase 2 with adults, and that actually we have a placebo arm in that, and we can see differences in the behavior and in the GI also with the placebo.
If you compare that patient age range 7 to 17 with your collaborator that did the study, and you looked at placebo from other studies or treatments, other treatments, your effects are different.
Yeah, our
basically.
effects are larger than the placebo effect that he's seen in other trials.
Okay.
Yeah, yeah.
Maybe a question about how and when the drug is used. So is the drug used when you see certain signs or symptoms of autism? And then the second part of that question is, do you envision the case where the drug could be used prophylactically instead of waiting to see certain signs that you just, as part of a treatment for people with autism, you just give the drug up front? And if so, when would you wean them from the drug, if at all?
Okay, so great questions. Prophylactically, everyone would have to take it, right? Because you don't know who's going to get diagnosed if it's prophylactically. That's diagnosis for autism is mainly behavioral, which that's another big need in the field. Biochemical biomarkers for autism would be great if we had a biochemical or a gene or a molecule that says you can select the population with this biomarker. We don't have it, it's all behavior. So it could be administered hopefully as soon as you have diagnosis, as long as FDA approves it, of course.
But in the case where you're looking for specific signs or symptoms to use the drug, right? So you could be diagnosed with autism, but maybe you haven't reached that threshold of treatment, right? Versus being diagnosed with autism and then just use the drug prophylactically.
Well, so at least on our first trial, we stratify by GI symptoms. So GI symptoms was kind of our definition of like, this is what you need first to qualify to be part of the trial. And so I'm not sure if I'm answering your question. So I think the focus was more on GI symptoms. And we can't just say this specific behavior is reduced with the drug. I know that with my studies, there's been a lot of my studies with autism also. One of the behaviors that is reduced a lot is anxiety. And so that's also part of what happens with kids with autism, that if you can reduce that anxiety, then a lot of the behaviors also improve. Yeah.
Okay, thank you.
And that would help also anxiety patients, not just autism patients, so that would be great.
One last question.
You mentioned that the innovation here is less about the drug and more about the treatment regimen.
It's together. It's the drug and the treatment regimen.
Okay, so I was going to ask if, are you aware of any other, has the drug itself been used before, or?
Not for autism. So there's similar drugs that have been approved for C. diff, Clostridium difficile, but not for autism.
Got it. But those other drugs would be the similar, same type of sample preparation and things of that nature?
There's little tweaks and differences, but yes, the idea is initially the same.
Got it. Thank you.
Great. Thank you so much, Rosie.
Thank you.
Great job.